everyone who's volunteered and put a lot of their enthusiasm into supporting climate change. Without these people, it couldn't have happened. And of course, we're also indebted to all of the participating chefs who've taken time away from their busy schedules at their restaurants to come and join us in the lecture here at UCLA. And in addition, our generous sponsors, company sponsors as well. So the scientific prelude for today's lecture, because it is associated with the class, Science and Food, the physical and molecular origins of what we eat. The theme is microbes, which many of you know have a pretty bad rep. But there are a multitude of benefits of microbes in society, ranging from health, the drugs that they produce, to biotechnology, the enzymes that they make even better than we can make them, and of course, food as well. Now these microbes, as many of you know, are very tiny, micron scale, which is why you can't see them all the time in soil or water or in the food that we eat. Notably, in our bodies, there's 10 microbial cells per human cell. So there's actually a lot more microbial DNA in our bodies than our own DNA. Microbes are also found in many foods around the world. And what they're doing there is producing a lot of different flavor compounds and compounds that alter the texture of our food as well.
All right, so one of the compounds that microbes produce that is found in many types of food is actually xanthan gum, which if you look carefully at the ingredients label on many of your food products, you'll see that it's pretty ubiquitous, and it really helps to change the texture of food and stabilize foods as well, such as your salad dressing. So you might be wondering, well, how does this come from a microbe? Well, there's a particular bacterium, Xanthomonas campestris, that actually secretes this
here at UCLA talking about food or even microbiology in general, so thank you for having us. Amy, I've known for a couple of years now, and she's been instrumental in helping Momofuku grow and further our knowledge about food. And one of the things that has been really interesting in the past sort of 15 years is the advancement in sort of the ingredients that cooks use. And it's become, I would think, a lot more, not difficult, but we need to know a lot more. Besides classical French technique or Italian technique or whatnot, the past 15 years, cooks have had to learn about all types of hydrocolloids, Jalan gum, carrageenan, xanthan gum, names that were very polarizing, ingredients that were very polarizing to the culinary world, because you're either in or you're out. You either wanted to learn more about how you could manipulate food, cook food in different ways, or you chose to take a stance where you didn't want to cook that at all. You were like, I don't want to cook sous vide. I don't want to know this. I don't want to add Jalan to, I don't want to make a fluid gel. I don't want to learn any of this. I just want to get perfect product. I want to make it beautiful. I don't want to learn anything else. I can say that because I was also one of those cooks that were like that until, you know, people like Wiley Dufresne and being introduced to people like Ferran Adria, they slowly sort of show you that there is a better way. You need to learn more about food. And microbiology, to sort of make it come full circle, was a way for us to sort of find what was most important about that question with this past 15 years of growth, 20 years of growth, was we were finally presented with the question of trying to ask why something is this way. Challenging the status quo of food. And that, to me, was the most important thing. I didn't have to use anything like called xanthan gum. But it was important because it started the conversation of how do I make things better? Why is this a certain way? And, you know, you just don't take for granted this is how you do it. And we really have been focusing a lot about fermentation at Momofuku, at the lab. This is Daniel Fell, who's the head of the research and development lab we have on 10th Street, a very small 200 square foot room, which is also one of the reasons why we're doing what we're doing. If we can make it in a 200 square foot room, we can do it anywhere. And we wanted to focus on things that were simple, the mundane, and investigate things that were already assumed that we already know how to do, like fermentation. I think there are a few cooks here. We all go to restaurants, we've all worked at restaurants, and they just say, this is how you pickle it. You do that. How do I make it better? They don't tell you. You just tweak it here or there, but we wanted to take a step back and figure out how to make things better. How do we make fermented products taste better? And sort of what we've been focusing on at Momofuku the past really 2 years, we've just been letting stuff rot. So, no joke. It's we're the king of rotting stuff. So, to focus sort of the universe of microbiology, because we are not really, we shouldn't be talking about this to you guys. We're cooks, okay? But we've really had not a struggle, but it's been a fun endeavor to learn more about this and to learn about things that I slept and cheated through in high school to relearn this. And so there's microbiology and microbes in food it's omnipresent. Microbes are omnipresent. So I wanted to sort of share with you one dish, and we could sort of illustrate how much microbes actually influence what we taste and what we eat. Okay. So this is a bowl of ramen that we serve at Momofuku. It's a hot stock, broth, pork belly, shoulder, slow poached egg, fish cake, seaweed, nori. So part of what we wanted to do when we first made this, serving ramen is, and when we were breaking it down to sort of teach you guys how much, where the flavor was coming from. I think that you're going to hear me say this word umami over and over and over again. And it's a recent sort of acceptance in the culinary world as a fifth taste on your palate. Salty, sweet, sour, bitter, and then umami, which is this mouth-watering sensation. It's meaty, it's a little it's delicious, right? And that's the action that we're trying to go after. And I find that to be the case in almost all the dishes at a restaurant. We're always trying to find that delicious factor, and we'll reverse engineer it from there. So we want to find how to make things delicious and layer that in a natural way. So we start with the broth, which is a very simple thing, but we layer it. We make a shiitake dashi. We roast bones to put in a tare, which is basically a chicken pork infused soy sauce. And besides all the things that happen on a microbiological level, on a, like sort of an atomic level, how we use noodles is really fascinating. We'll have a special guest appearance on the slide to help explain something that is really not that well known in terms of alkaline salts and how that affects noodles in hot broths and stuff. And basically tell you how we try to make this delicious. And this is very much an oversimplification of what I'm going to try to say today. Microbes produce enzymes. Enzymes develop amino acids, which are glutamic acid. Some of them are glutamic acid and aspartic acid, which is sort of like the middle child of the amino acid family in this, because it always gets overlooked. But we're always trying to make umami. And these are two pillars to making things delicious, things that we eat all the time. Whether it be Parmesan, whether it be soy sauce, miso, Doritos, fast food is loaded with MSG. And it's something that is a touchy subject, taboo subject, that hopefully we'll be able to illuminate a little bit of light on. So going back to the broth that we make for our stock and ramen, the first thing and the most important thing is making a kokudashi. And kombu is a kelp, but it is very high in glutamic acid and aspartic acid. And one of the important things about it is we cook it at 60 Celsius. But this number is something that's a good example of something that's sort of happened over the past few years that's become accepted to cook dashi, something that the Japanese and various Asian countries have been cooking kombu for hundreds of years, but nobody, until very recently, the past 20 years or so, like the mid-90s, did people figure out, well, maybe there's an optimal temperature to cook the kombu at, and it happened to be 60 Celsius. But people started to ask the question, is that even the right temperature? So even now, it's up for debate, what is the optimal temperature? If you cook it too high for too long, it gets a little bit bitter and a little too overpowering and cloudy. And 60 Celsius seems to be the one temperature that there seems to be agreement on where it extracts the most, what you're after, which is the glutamic acid. So we cook it at 60 Celsius for like 60 minutes, 90 minutes, and we then add shiitakes, which is sort of this magical item. We hear about umami a lot, and one of the magical ingredients that Mother Nature makes for us is shiitake, and mushrooms in general, but especially dry shiitakes. It just has this amazing, I don't even know, I think it might be the food with the highest amount of glutamic acid when you add it with kombu. It's very high in this thing called guanylate. And Dan and I have sort of been messing around with basically the shiitake essence. And we don't even know how they make it. We bought it at some really random Chinese supermarket. And it says not MSG. It's so delicious. It's called guanylate 7. We have no idea. We try our best to find information about it, but guanylate is something that, again, it's similar to glutamic acid, but it adds so much flavor. It is extraordinarily potent stuff. And, when we add it to just about anything, it immediately makes something taste that much better. This is, katsuobushi is probably the one item in Japanese food that is, I believe, one of the true foundations. Traditionally, it's made with bonito, a type of tuna. And it's steamed, smoked, dehydrated either in the sun, and inoculated with a mold called Aspergillus oryzae. There's two types of making katsuobushi. One is through heat, changing it to a lower temperature and higher temperature, and one is inoculating with Aspergillus oryzae. And that is the Japanese called koji. And my fascination with koji and katsuo has really shaped what we do every day at the lab. And I made Dan Felder's life basically a living hell because he has to deal with some type of katsuobushi every day. And it's a very simple thing. It's a petrified piece of fish, basically. And it is very high in glutamic acid and things that just make, again, umami. And in combination with kombu and shiitake, it gives us that sort of base to make that great ramen broth that we're after. But it's a really fascinating process to make. It's something that we've been replicating, and I'll talk a little bit about it a little bit later. And this is Aspergillus oryzae, if you see over there. It's a mold, and scientists have traced it back from like I don't know, was it a million years ago? I can't remember. To Asia. And without Aspergillus oryzae, we really wouldn't have Asian cuisine as we know it today. It was responsible for soy sauce, sake, miso, basically all fermented products. And it gives us the flavor. And that's where people don't realize, myself included until very recently, that microbes equal flavor. Not all microbes are bad for you. In fact, a lot of microbes are responsible for all of the delicious food that we eat on a constant basis. So, as you can see, you take a raw piece of fish, you do a few things to it, and it turns into a petrified piece of wood. And a really good katsuobushi, if you crack it open, has the texture and it looks like amber. It's very hard, and when you shave it, you get these leaf-like shavings, and it's beautiful and delicious. So, right now, actually, if you guys came in, we gave you two little samples of stuff. One is the white powder. It's MSG. If you are allergic to MSG, or you think you're allergic to MSG, something we'll talk about later, just don't eat it. But I don't want to sound redundant and trying to go over and over and over again. What we're after, that action is we're trying to replicate that flavor that you're eating right now. Right? This is on top. This is MSG. This is the structure of MSG on a molecule level. Right? This is glutamic acid. This is the action. This is everything that we're trying to create when we make that broth and all the other ingredients that I'm going to talk about in that bowl of ramen. This is also glutamic acid, the same acid you're going to find when you dry age beef, you eat soy sauce, you eat, Parmesan cheese, tomatoes. Your body digests it the same way if you are eating MSG right now, right? And this is a sodium molecule. So the wonderful food scientists at Ajinomoto figured out the best way to disperse glutamic acid and to cook with it was to add a sodium molecule. So it's basically one sodium molecule added to the glutamic acid chain, and that's what you have. You have monosodium glutamate. And it's just an artificial way of adding what we're trying to get in a natural way. So the argument of what's natural, what's artificial, that is something that I would like to talk about later as well. Oh, this is my favorite, by the way. This is a giant tub, massive, it's like this big, of MSG. So, you got the broth out of the way, we make the toddy, so we get soy sauce, which again has a lot of glutamic acid. We want to infuse it with, chicken bones and pork bones, and, that's our job as cooks, is to not throw anything away, to be as frugal as possible, and to use every scrap to make something delicious. So, again, that's why we make stocks, but instead of making a stock with water, we're basically making a fortified stock with soy sauce. And we're roasting the bones to get Maillard reactions, which is a caramelization of proteins. And in that process, you enhance the glutamic acid content. So when you brown meat, you actually make it more delicious. Again, called a Maillard reaction, and we're trying to add that to, again, another way to add umami. And we sweeten it with a little mirin, with a little soy sauce to balance out the flavor. And that's how we season the ramen. Soy sauce is something that we've been making at the lab. And again, you take soybeans, you cook them, add water, salt, inoculate it with Aspergillus oryzae, koji, and you wait 6 to 9 months, 6 to 12 months. Our homework has found that it was probably developed to sort of stretch out salt. Salt back in the day was extraordinary valuable commodity. And this is, you don't have to use that much salt to actually produce something that is delicious and has a lot of savory, salty characteristics. Sneak peek of the man. So this doesn't really deal with microbiology, but it sort of does, because again, this whole concept of microbiology is something that it's about looking at things that are very simple to the naked eye, something that you would just pass over. But when you really investigate it, it's deceivingly complex, so complex that we really don't know that much about it, particularly alkaline salts. You see it in America with pretzels. You spray pretzels and it has that sort of that flavor that is so distinct to pretzels. But in Asia, it's used primarily in hot noodle soups, and it gives it it's sort of magical. Like there's an ion shift, and it changes the temperature of the gelatinization, so it's chewier. Basically, without it, we wouldn't have hot broth. We would not have ramen because it would just disintegrate. And I will have our special guest, Harold McGee, explain.
So we might start with the question, what is alkalinity if we're going to be talking about alkaline noodles? And alkalinity is the opposite of acidity. Alkaline ingredients are the unacids in cooking, and they have to do with the nature of water. Well, it turns out that most foods have a lot of water in them, and so the nature of that water and the balance between the hydrogen ions and the hydroxide ions makes a big difference to the way the other components of the food behave. The carbohydrates, the proteins, the fats are all very sensitive to their chemical environment. When you add alkaline ingredients to a noodle dough, you change the chemical environment for the flour. The color changes. The noodles get yellow. The texture changes. And this is not that well understood actually, but somehow the interaction of the gluten proteins is changed in such a way that the noodles become much firmer. And there's the effect on flavor. The flavor of alkaline noodles is really distinctive. Flavor is the aspect of food that we know least about. So one of the great things that alkaline salts do in a noodle dough is to make the noodle much stiffer, and therefore it holds up better in the soup. You know, if you take an ordinary noodle and you put it in the hot liquid, eventually it's going to dissolve because the proteins are just going to fall apart into the liquid itself. So that's probably a big part of the reason why alkaline noodles have found a special place in soups.
St. McGee, one of the reasons why a lot of cooks are doing what we do today is because of the work that Harold McGee's done, and he's been a great asset to us. And he was actually the first person I called, or I sent a letter to be like, how does alkalinity work? I don't understand it, it's magic. And he basically said something along the lines where I still don't understand today, but you know, you add alkaline salt, katsuo, there's a, that's the Japanese name for it, but it's a bicarbonate salt, and you change the pH level, it raises it. And really just changing the pH level of the water changes this dough completely. You can make a pasta dough with just water and flour, and you can make a side-by-side comparison with just water and like a certain percentage of katsuo, and it's completely night and day. One dough is extraordinarily difficult to work with, and that's the one with katsuo. It's so hard, and it does turn yellow. If you add too much, it turns a little green. But it has this texture that the other dough doesn't have. It's resilient, and again, like in Asia, they eat soup so hot that it would, again, disintegrate the noodles if it didn't have it. So while it's not technically microbiology and what we're talking about, it's again something that to the naked eye is, not that sexy to talk about, but to me it's extraordinarily interesting. And the better we understand how to make noodles, and going back, basically, I don't know why people don't use it more. Why is it just only in Asia? And trying to make Italian pastas with a farce with no egg yolks and stuff like that. There's endless possibilities if we explore just a little bit more. And these are some of the things that we add into ramen, and you might see it quite a bit. There's endless variations. We have pickled vegetables, nori, again, seaweed is full of umami. We add pork. Miso, again, is so high in umami, and actually so is an egg. We slow poach it. And the flavor that we're trying to go for in our ramen was not that it's Americanized, but we're trying to go for a flavor that is American. Like, it's very, very common right now in Japan to make a very seafood-forward broth. And I wanted something that was a little bit smoky. That's why we had bacon, and we had all these things that, again, in retrospect, I can see that it was just layering umami. And that's, again, what we're always trying to get at. Growing up in a Korean household, I ate kimchi with everything. When I like to eat ramen, I like to eat it with kimchi. And going back to some realm of microbes, it was a shock and a revelation to me when we did some more homework on just how kimchi fermented. And for just about 6 years, I told every cook that worked at Momofuku the wrong thing. I said the fermentation happens because there's proteins from the shrimp or the oysters that we introduced to it. I was dead wrong. And it's something that was even so cool that I couldn't, it's so simple that my mind is, I get excited just talking about kimchi, so just one tongue. The fermentation is a lactic acid fermentation, and it's almost, if somebody told me that kimchi is exactly like sauerkraut, I'd just laugh in their face, right? I was like, kimchi's obviously more delicious, right? So in both sauerkraut and kimchi, you salt it. You salt the cabbage. And that is actually the process that changes everything. When you salt the cabbage, you change the environment in terms of where the cabbage lives, and it prevents anything else from surviving in that environment. And what is really fascinating is that in cabbage, already present in like a Trojan horse almost, are 3 types of bacteria that already live in cabbage. Whether it's snapper cabbage, Savoy cabbage, it's pretty abundant in it. And when you add that salt content, it creates a perfect environment for these bacteria to thrive. And they begin to eat the glucose, the natural sugars in the cabbage, turns it into lactic acid. Lactic acid turns it into CO2, which facilitates the entire fermentation process. There's nothing to do with anything that I've added outside of the salt. Everything else is just flavor. And just dwelling on that, it really changed the way I look at food. I was like, wait a second, this is something that I've held for years as accepted fact, which is totally wrong. And no matter what I do to make it better, I had to relearn how to make kimchi. And there was a reason. I like my kimchi to look a little bit more high salt content. Which I feel like, because there's effervescence after the fermentation process, I thought it was the bubbles that made the kimchi taste different. No, it was because there was more action going on a microbial level. So kimchi is a sort of a wonderland of microbes and bacteria, and again, it's very simple to do, but it's just amazing how it works. just add salt and you're going to have a lactic acid fermentation. So, you know, having covered some of the ramen and some of the things that have microbial effect in the flavors, we've been working on at the lab different techniques. How do we introduce more microbes? How do we expand on microbes to make more flavor, more delicious food? What are our new techniques that we can, or not even new techniques, old techniques that we can analyze and sort of tear apart like we did with kimchi. One was going back to katsuobushi, which is, again, a fundamental, one of my, I think, a genius product right next to like puff pastry. It's one of these things where I just wanted to see if we could recreate it. And we don't have access to bonito. And one day, about 3 years ago, I decided, why don't we try to make it out of pork? Because I hate tenderloin. I just hate that. It's too lean. It's sort of not roasting. I just don't want to eat it. But when I was looking at this tenderloin, it looked so lean. It looked like bonito. And just randomly, I was like, why don't we just steam it, smoke it, and then I'll bury it in rice, raw rice, which is what I did. And it was like this technicolor weird thing, yellow, green, purple, all these things. Every day I would peek in. And it was, I was like, oh my God, like, am I dying because I'm breathing this? Like, what's happening? Somebody had to taste it. And I shaped it on katsu. I shaped it, and this is what you get. You know, it was eerily similar, almost exactly similar to the texture of amber and really good katsuobushi. But here we made it, you know, 9 months after the fact, not using fish but pork, and you get these beautiful shavings that looked just like katsuobushi and had a flavor that was reminiscent to Japanese katsuobushi. But here we are, we made it with American pork, New York, Microbes, that terroir that is literally on 10th Street. And I had to ask myself, what did we just make? Is this Japanese or is this American? It doesn't matter. It didn't matter to me. All I knew was we had stumbled upon something completely by accident, and it seemed to work. It was such an accident that we could not replicate it. So we had to enlist the help of Harvard University, Rachel Dutton and Ben Wolf, 2 microbiologists that work quite a bit with cheese. We sent them a sample of it, being like, we can't make this again. What can we do? So we made it over and over and over again. Because it was one of these totally lucky things. I don't know how the hell it worked, but it worked, right? We created a pork bushi that was looked and tasted something similar to tuna. I mean, to bonito. So we would glue together using transcontaminase pork, steam it, smoke it, and let it rot with Aspergillus oryzae. So again, it's inoculated with koji. And this is what happens. It gets this beautiful white color, and it just grows. We can now figure out how it was made, because we actually didn't inoculate with Aspergillus oryzae. I actually forgot to do that step. What actually happened was, something else happened, and we stumbled upon it completely by accident, and it was called pichia, which is another type of mold, traditionally found on fermented, cured, smoked pork products, like ham and sausages. But what was interesting was we didn't really realize how important this sort of discovery was for us, because it created in the exact same fashion what Aspergillus oryzae koji does, but we didn't use it. We used literally a microbe that was indigenous to the corner of 10th Street First Avenue. And you know, you start to ask the question, like, then what necessarily is this? Because it's very important. Even if we make it with Aspergillus oryzae, it traces back to Asia, right? No matter what flavors we do, it's going to always originate from Asia. So if we make you know, there's billions and billions of different types of microbes out there, and we stumble upon one there's most likely more, right? And it acts in the same fashion to create a very similar product to katsuobushi, but with the different flavor profile, which is the same reason why you have terroir in wine, right? There's not that much difference in terms of how you make wine. It is the soil, it is the air, the water. Really, it's the microbes that affect and change how one Pinot Noir in California will taste completely different than the Pinot Noir in France. So in that sort of same vein, we realized that we were trying to make American food or New York food. We were really trying to discover how to make something that wasn't necessarily Japanese. It's paying homage to Japan, the techniques we discovered in Japan, but trying to make it for us an American product. So we really wanted to sort of just make everything raw, and that's what we've been doing. And this quest for everything raw is something that Dan Felder is going to tell you a little bit more about in terms of koji, and our miso production, and soy. Dan?
So basically, at a point when we had been working on the bushi, and Ben and Rachel at Harvard were, tutoring us in basic microbiology and sending samples back and forth, we started to look around at what else we were, taking for granted. What other products were we using every day in our restaurants and not fully understanding? So, an obvious place to start was koji and miso. Koji is essentially the sourdough starter of miso and soy sauce. So it's a grain. Traditionally, it's rice. The first evidence of Aspergillus oryzae was found in rice patties. For us, we do a barley koji and a basmati koji. Just in terms of our space and limitations, they seem to hold up the best. So basically, the grain is steamed and then inoculated with Aspergillus, and it goes into an aging chamber from 36 to 48 hours, at which point enzymes start developing, such as amylase, which is converting starch to sugar, and basically that's your starter for the next step. And this is what happens when you don't pull it out, when the mold's still white, which is sporulated koji, which is also where microbial evolution comes into play. That's basically traditionally out of every 10 batches you would let 2 go to spore. And that signals basically all available food is gone, so the mold goes into a reproductive phase, which looks like this. So this is time-lapse over 36 hours of Aspergillus colonizing. You can see the hyphae start growing, and then they conidiate, which is the balls around the end, and ultimately those will turn colors, and the normal colors you associate with red, going moldy, signals the reproductive phase. So basically at this point, you can harvest these, and that's also how they're so similar to a sourdough starter. They continue to evolve. They're living organisms, so generation after generation will pass down spores, and they develop their own characteristics and nuances.
Yeah, well, and again, Dan brought up a great analogy. It is a lot like sourdough in terms of if you can understand the nuance of it. But our understanding of microbiology was so non-existent that the wonderful people at Harvard literally had to draw cartoons for us to understand what the hell was going on. Trust me, Dan didn't sound like a science teacher 2 years ago. And, we really had to work hard to understand what the hell was happening. And we still are learning and don't know that much about it, but we're very fortunate to work with really people that are so much smarter than us. So,
thank you.
Drawings like this. I'm not even going to tell you what's going on. We really, I can't tell you, I think they were dumbfounded just how little we knew about cell structure and enzymes and amino acids. I was like, I don't know this stuff. no matter what I read, I didn't get it. So it was really very helpful for them to sort of tend to our 6th graders, and it was great. And this is actually what we do at the lab. And you can see how small it is and what we do, and this is, you know, Dan and Veronica, who's up here from New York City as well.
So this is basically the process of making miso. So you can see it's 3 ingredients: koji, the primary medium that you're fermenting, and salt. And salt represents the safeguard and basically the ability to ensure the environment is optimal for you know, the enzymatic conversions to happen, the development of yeasts. And beyond that, the main variable that we screwed up over and over and over again was the environment. And it's really vital both for making koji and ultimately aging the miso. We use quite a low salt percentage, which facilitates incredibly, and a high koji percentage, which facilitates incredibly fast misos. So instead of looking at it in years, we look at it in days. The miso that you guys will taste later was aged for 5 days. So basically it's packed, weighted with an equal volume, and then it goes into the aging chamber between 35 and 42 Celsius. Up to 42 is kind of the faster it will go. And you can see evidence of how many times we blew it. Just trying to get this going.
But that's the funny thing is, the more we never thought we'd take a scientific approach to making anything. We realized that the scientific approach is making error after error after error, and realizing that's not the way to go, so we'll go this way. That's not the way to go, so we'll go that way. And over a 2-year period, we're now in a process where we can make very reliable misos, and we're not just making it out of soybeans, we're making it out of all sorts of grains and starches. These are, miso is the Japanese version, gang is the Korean version, kishio is Chinese. So, fermented soybean is obviously a staple in Asian cuisine, and that's something that we want to produce. Just not only for our own application, but we want to make it for the restaurants. We want to make stuff that's not just soybean based, and we wanted to get an understanding of koji, which again explained, but we were so excited about Pishia that we realized that we needed to learn how to walk before we run. And Pishia is something that is, fascinating, but we don't know anything about koji. So we just sort of jumped right in and tried to learn as much as we can about this wonderful mold called Aspergillus oryzae. So until we get to know what we're doing with that, we're not going to really focus on anything else. And to sort of help reinforce the notion of microbes in food and how it creates flavor, it's, when we're making miso, in a weird way, we're after this, dry aged beef. Your dry aged beef, you're putting it in an environment that's going to create, it's going to be a magnet for all types of microbes, bacteria, mold, and all of that stuff over a period of time in a perfect environment for dry aging beef is to, one, make it a little bit more tender as things break down, cell structures, whatnot, but most importantly, you're dry aging beef to make it taste better. And what you ate, MSG, is the same thing that you're doing when you're dry aging beef. So in a way, it's a little bit an argument of semantics, so that's the way I look at it, right? This is the mold that creates our lattice work. It's actually quite beautiful under a microscope, but because we don't know exactly what we're doing, I mean, we know what we're doing, but we have to compare it to other things that we do understand. We do understand dry aging beef, the concept of it. So it allows us to sort of make misos with a little bit more confidence. Other things that we're making in the lab are, hams, bacon. These things are so high in umami. And, right now we have rabbit. How long is the rabbit in there?
We're doing between 7 and 20.
It's got a really strange funk to it, and
It's really good.
I don't particularly love it as much as Dan loves it, but I think it smells gnarly. I don't want to eat it. So, but something that does smell funky and is delicious is hams, whether it be from China, whether it be from Spain, whether it be from America. American country ham, I'm more partial and fond of. There's a lot of action going on in terms of microbes, and people don't realize that hams and salted pork products are really fermented. It's not just curing. There's over a period of time, it's transforming itself through a stage of fermentation, and that's why the hams taste so good. Not just because you're adding salt. Because I worked for a chef once, and I was like, why? We're making our own prosciutto. He's like, what's happening here? He's like, oh, it's the salt. It's curing it. Okay. How does that make it taste better? He's like, it's the salt. Don't worry about it. It's not good enough to know that anymore. And as I said, starting out this lecture, you have a division of a group of chefs and cooks that don't want to know anymore. And this is the most basic level of cooking. Fermentation has been around for thousands of years. If we don't know how to analyze this and start to ask the question of why and how do we make it better, this is now, and I don't think you can avoid it. And this is what I want to force upon just not only my cooks, but my peer group to start to ask, how do we make fermented products better? Because every restaurant's making pickle, every restaurant's doing in-house charcuterie. But if we don't have an understanding of how the process actually works, we're just making the same thing over and over again. And I think it is our responsibility to make things better, or at least know how to make things better. One of the things that we've done is, if you come to the lab, you'll see that it smells really funny in there. Part of the reason is these guys. We're making, how many days has it been?
2 that are 5 months, and then 1 that's fresh.
Yeah, so basically we took fish, we chopped them up, we saw one with guts intact, one without the guts, added a high amount of salt, and just let it rock. And the smell is extraordinary. And, but people add fish sauce all the time, right? And we're trying to make our own fish sauce. We're trying to better understand how to make better fish sauce. If we understand that, we can incorporate it into different ways. And maybe we can start to, this is something that I like to think about, is like garum is part of Roman cuisine, right? Were there dishes, back in the day, did it taste like Southeast Asian food? I have no idea. But garum was, and probably did taste a lot like fish sauce. It's the stuff that we're making. So you start to wonder about flavor profiles and what is delicious and how it was delicious throughout the years, throughout history, really. Different types of amino acids, boring. But this is Dan Nathan's fish sauce.
For this fish sauce, we actually, in light of having learned about koji and learned about miso, we started adding koji into our fish sauces. The idea being that with the addition of the protease enzyme, that we could facilitate faster fermentations and further amino acid development. So now that we have some basic knowledge over the past over a couple years of making katsuobushi, familiarizing ourselves with koji, making fish sauce, really basic things, we realize just, again, how complex it is. But we have now a framework and a foundation in which we can now use these techniques and apply them to other ingredients. One is pistachio miso, and if you would please be kind to taste the other ingredients. Can you guys hear me? Yeah, better? Okay. What do you guys think? Delicious? Yeah. You're still higher, man. Sweet. How many, I just want to quick hands. Who didn't
You guys basically ate the same thing. Right? You have to ask yourself, why the hell didn't you eat the MSG? Right? Your body's now consuming the miso, which is just as high in percentage of glutamic acid as the miso. Right? Now you have to ask the question, why the hell didn't you eat it? So I'm not going to go to where I want to go. You have to ask yourself, again, not only as cooks, as consumers, ask the question, why? Why do you think there's Chinese restaurants in? Why do you think MSG causes headaches? When all the studies suggest and that there is no such thing. It's psychosomatic. So there might be people that are sensitive to MSG, but as you guys can see, do you guys, anybody have symptoms of Chinese restaurants? Anybody? No, right? And I've seen it too, and I've heard it too many times. When we have an open kitchen, I can't eat soy sauce, I can't eat Japanese food, I can't eat Asian food. And you know what? 2 hours later, they're eating at Bavo, give me more Parmesan on my tomato sauce and my pasta. It's the same thing, right? It's really the same thing. You're eating glutamic acid. That is what is delicious, and that's the action that we've been after. Momofuku, it's the same thing where I think we need to do some more studies and asking the question, why are people so adverse to MSG? I don't want to cook with MSG, but maybe it's completely unnatural, but maybe it's not unnatural. We cook with one gum, we cook with modified starches, we cook with xanthan gum, the same technique. And we take that miso, and we've been very fortunate to not only have Harvard help us out, we've had New York University help us out as well. And they're my team of microbiologists have let us, and Dan used their lab, and we spun it in a centrifuge that has a lot more RPMs than the one we have at Ssam Bar, and it's separated into 3 amazing products. One was the miso that we're eating right now, a pistachio oil that is so elegant and delicious and light. And the other one is basically a soy sauce, tomorrow. We made a gluten-free, basically, soy sauce that has subtle notes of pistachio, but for the most part, it's one of the most delicious things I've ever had to taste, literally. And when Dan came back from NYU with these things that were spun in their centrifuge, it was we were little kids on Christmas Day. It was not much fun. We're like, oh my God, what can we do? It's so delicious. You see one with chickpea. The chickpea tastes like Parmesan. It really does. So there was a lot of fun. Pine nut miso is something that is actually my favorite. We didn't bring a lot of it to you. We just want to give you one because it seems like most people seem to like the pistachio miso quite a bit. But now we're making misos that aren't Japanese. They're not soybean based. We're making it with American products, really local, whatever, that's how you're supposed to anyway, but it now changes, it now adds to our repertoire, adds to our arsenal, it now adds not just flavor, but we're able to introduce new ways of cooking food to the customer. One of the last things to talk about is vinegar. Again, I've always been fascinated with vinegar, maybe because I don't know how Modena, everyone drives a Lamborghini because they sell, I don't know, balsamic vinegar is crazy to me, but, if anyone's been in Modena, you'll know what I'm talking about. I wanted to make vinegar because I don't believe we make good vinegar in America. And everybody wants to make great wine. I've said this before, I want to make great vinegar. I want the rotten stuff. And one of the things we produce great in New York State that California, I believe, doesn't have an advantage is apples. And don't start growing them there. Because we make great, I think our apples are fantastic, and we're making beautiful apple juice. And it's what I've seen in Europe when, some beautiful vinegars in Italy and France. And what they're making is actually a great wine. You can't make something delicious out of crap. Yes, you can, but, that's another story. But it was a revolution to me, just like when I went to Japan to see how shiro miso was made in Kyoto, to see how wine great wine was turned into vinegar. And they weren't even selling wine. They were making great wine to turn it into extraordinarily delicious vinegar. Something that is completely overlooked, just like microbes. And we wanted to make that with apple juice. And Dan can explain a little bit more.
Yeah, so basically in the same vein as learning about miso, it was how do we understand a basic concept and then build on it and innovate in ways that, things that haven't really been done before. So we started with apples, like January in New York, it's apples. So basically we learned how to make apple wine. So as opposed to doing a cider vinegar, we would treat it more like a mead and using natural sugars to then create the first step of the two-step fermentation that creates vinegar. So the first step is a yeast conversion of sugar to ethyl alcohol, and the second is a bacterial conversion by native airborne bacteria to acetic acid. And basically, the second stage is aerobic, and the way that we speed it up is by using an aquarium air pump. So what we have here are two vinegars that we're making for Co right now. One is an orange Riesling and a cherry Riesling. Basically for this we took the Riesling that's on the very famous foie dish at Co.
Very famous?
I don't know. Very notorious. And use the air pump to do both an infusion and a conversion. So this is an orange where basically we can control the bitterness by using the air pump and then removing it before it gets that pithy, bitter flavor.
So Dan and Veronica, on top of the rotten fish, the rotting grains, we have rotting fruits, and it's really something else. I wish I could capture the smell of the lab for you guys. So some of the other things that we're working on outside of vinegars, some side projects. Dan's been making his own camembert cheese, and it was really delicious. Congratulations on that, one. What we learned with Pishia, going back to katsuobushi, and you can sort of see how one thing sort of has this domino effect in terms of how we make everything else. With the more knowledge we gain, we can sort of apply that to other things. Again, like the pistachio miso, we wanted to see if we can make katsuobushi out of something else other than pork. We tried chicken. Sean Brock and McCrady's just did scallop bushi, and we actually made this a very successful attempt at chicken bushi, tori bushi, but we actually didn't use Pishia either. We didn't use asparagus. We inoculated with camembert mold, and we got the same result. And it tastes, I don't even know how to describe it, because we've never had anything like this before. There's nothing to compare it to. Dan, can you explain this?
Yeah, so this was kind of a side project that was started by Veronica, which basically she decided to make koji powder out of our barley koji, because it's this amazing, rich, funky, dry-agey soy flavor. And we were using it combined with sea salt as a finishing salt on dry-aged meat, because one of the sad parts of cooking dry-aged meat is that you lose that primal thump that it has when it's raw. So we combined it with salt and have been curing lardo that ends up getting all that funky, moldy, dry-aged flavor in a medium that really isn't associated with that, or you can't really age that way.
Because fat equals flavor too.
Yeah, fat equals flavor too. And it ended up being incredibly delicious, and it's one of those moments where we cast a wide net about what we're learning about, and then have the opportunity to kind of smash the ideas together, make new stuff.
We're making soy sauce, barley soy sauce. It's fun. There's all sorts of stuff. I wish we could have brought samples for everybody, but the soy sauce takes several months to make. The miso takes 5 days, so I apologize. So the microbial future. You know, there's a lot of, we talk all about microbes, and we are most fascinated with this guy right here. This is what we really want to sort of bite into once we figure out how to, exactly, not master, but have a better understanding of what we're doing. And this is called Neospora. So when we're in the process of figuring out if there's any other molds that were similar and acted in a similar vein to koji, we started to literally cook and rot just about everything. Every square inch of the lab was a dish of something rotting, and we sent it all to Harvard. And
Dave's name on the return address.
yeah, and you know, Rachel and Ben were actually quite upset with us because we sent them a lot of samples, none of which was, you know, it yielded any results. And then, you know, we were a little bit heartbroken, and then we got a call or an email the next day because there was like one sample left, and it was this. And they didn't, because this thing was crazy. The mold grows so fast, it's insane, as you'll see in a second. And we didn't know what it was. They told us it was Neospora, which is very common amongst, I guess, DNA analysis of genes. But nobody at the time realized that it had flavor. And again, microbes equal flavor. Mold equals flavor. This tastes delicious, believe it or not. You know, and when it just starts out, it tastes a little bit like bubblegum. And how many hours?
36.
This is in 36 hours, this time lapse. And it grows so fast that the tops of petri dishes were like, you know, falling off. So when it gets around this stage, it has this apple cider-like flavor. And it's something that, you know, we're dangling ahead of us so we can sort of get there. We want to figure out again how to do this stuff. But again, microbes are equal flavor, different microbes are equal new avenues for us to go down. And you know, we thought we had discovered like a constellation or something, but the reality was we didn't discover anything, because in Java, this is the liqueur they make. It's called oncom. And I think Rachel figured out, anyway, someone figured out that they chew peanuts and they spit it out and they inoculate it with Neospora to create the alcohol called oncom. And I haven't tasted it, and we actually haven't gotten to the bridge where we're going to make that ourselves. But you know, it gives us ideas, and you know, that's pretty much where we're at. And it's been a long journey for us. I actually don't get tired talking about this, even though we're not experts. We wouldn't be able to do this without the help of Amy and the team at Harvard and NYU and everyone else. Like, this is something you just don't do at home. And you know, you can't make miso, excuse me, but some of the other weird stuff that we're doing. But I guess that's it. Thank you guys. That question. No.
I mean, with say 200 kinds of kimchi that exist.
Well, yeah, I mean that's the funny thing. Reverse engineering in a way that what should I say? You know, there's a friend, Chef Magnus Nielsen in Fabriken in Sweden. He's made his own soy sauce without aspergillus as well. And it's a flavor where you're like, okay, now it is an issue of semantics in a way, like what is that? And if we all made the same soy sauce, if we all made the same sauerkraut, we had the exact same cabbage, but we made it, and we took it back to 50 different places around the world, it would taste completely different. And I think that, you know, we haven't crossed that bridge yet, but we're actually trying to, you know, there's a lot of chefs around the world that are working on the same project, and we're building this sort of database that's Maybe it would exist, but I don't know where it is. So.
Have you figured out what the ambient flavor of bacteria in Penn Street is?
No, I mean, we call it microbial terroir just because it sounds funny. But that's the thing, is it's constantly changing. And we have no idea how we stumbled upon quiche. We didn't inoculate it with it, it just happened to be. It could have been that the mailman dropped off our mail and he was eating a ham sandwich before. Like, we have no idea where these microbes are coming from. So it's there's a lot to know. There's a lot to actually discover and learn about.
Here's an excellent question from the audience. How do you not poison yourself? Corollary tastes first.
We give it to Dan to try first.
A lot of it, in terms of safety, was about, understanding what we were doing, and each of these fermentations have specific safeguards to prevent pathogenic bacteria, to prevent unwanted microbial activity, and so we do those things, and then we send it to Harvard.
I guess there was a Harvard graduate that works for the NIH or Wall Street Hospital, and he's like, you guys are dealing with extraordinarily dangerous stuff. You cannot taste this on your own. So, we have a good idea. It's like making yogurt at home. That's the way we're looking at it now. And that's why we're not working with Pichia. We're not working with Neospora yet, because we just don't know. And until we get the green light from professionals, we're not going to venture without proper guidance. And we're constantly in communication with them. So,
I had a friend, a former colleague at the Times, who was an expert on the cooking of 10th century Baghdad. And one of the condiments that was really famous back then was made from rotting barley. And one of his scientist friends told me that is an excellent way to create aflatoxins, which are about as carcinogenic and substances as exist on the earth. So he took one little taste of it and spat it out 5 seconds later. Then another scientist told him later that basically what he'd done is create soy sauce, so it's hard to know.
it is hard to know. So, we don't know. So.
The question from the audience. Have you done collaborations or thought of collaborating with the Momofuku Lab or the LOE Lab?
We call it Momofuku. Dan's actually going to work with Mars. I know Renee and Lars spoke at UCLA a couple weeks ago. And we're how often we speak? A lot.
A lot. Lars and I, for each of us, the other person was the first person we'd ever met who'd made miso or done any of these things. So there's a kind of camaraderie and collaboration that, competition in terms of who's developing what and how are we going about it. And it's been an incredible motivator and catalyst for our work, collaborating with them.
And it's fun because it's healthy competition, but everyone's helping each other out. As I said, everyone from Daniel Patterson at Akoi, Sean McCready in Charleston, Renee and his team in Denmark, and Magnus in Sweden. Everyone's sort of sharing their discoveries, and it's a fun time to be messing around with this stuff, at least on the cook side.
With some of the other chefs that you've mentioned, the sort of experimentation is almost manifest on a plate. I mean, you can see how they went there and there. How does what you do in the lab end up on the plate on Saturday night at Momofuku?
I would think that because it's so much closer to Ko, that's where we family meal. There's just Dan used to work at Ko as a cook, so there's a lot more synergy there than the other restaurants. But because it's a 12-seat restaurant, we can sort of work on the ideas and have it, you know, we actually haven't put anything on the menu from the lab yet, but, you know, it actually helps a lot with the creative process and coming up with dishes, because all of a sudden, you know, I always look at sort of cooking as like a Crayola box of crayons. So instead of now having 6 colors, we can now give them 12 colors, and we're not going to, you know, send anything out until we actually are very familiar with that. So it's fun to see the creative process with the cooks and Dan introducing, say, the miso or the tamari. We were recently messing around with a flute dish, and you know, just obviously that's something that we were initially going to first do. I think that was the first thing we did. We sliced raw fish and we put the pistachio tamari on, and it was a revolution to us. It was a revelation. It was unbelievably delicious. So you know, it's a slow process, but it helped the creative process.
Do you ever, does the health department ever look at your room of gold?
I don't know what you're talking about. This conversation never happened. They don't exist.
Okay. Peter, this question's for you. You have a fan who says the Momofuku book is such a great read. How is it collaborating with David, who is unusual?
That's almost generous. There are many types of unusualness to David Chang, but no, I mean, it's like collaborating with him. You know, we fight a lot, and then we make stuff. I don't know. Speak up.
Speak up.
He's just mumbling anyway.
No, I mean we had a really, I don't know, we fight constantly, and that's how we collaborate. You know, we find things that we disagree on, and then
Yeah, I think people don't realize that we actually when we say we make fun of each other, we really just try to hurt each other's feelings. And then out of that process comes out, you know, Lucky Peach or something. It was, you know, Peter is always like, that's so dumb. And I'm so stubborn, I'm going to continue to do it until Peter accepts it.
I've been thinking that their whole lab rotting thing was really dumb until tonight. And then there's this constant competition with Dave and I to like be right, and I realized that was wrong tonight. So I found this presentation very deflating.
No, but it is a, you know, I wouldn't say it's sort of like brotherly, but it's a competition to prove each other wrong. And when we're proving each other wrong, we literally are making fun of each other and trying to hurt each other's feelings. So that sort of forces us to sort of come up with something. You know, I just read the recent, you know, the draft for the next Lucky Peach, and I was really telling Peter how much it's going to suck, and it's going to be this, it's going to be that, and he proved me wrong. And I think it's really great. And the less work I do on it, the better it seems.
Speaking of Lucky Peach, some of the audience, one of the esteemed students, said he read the ramen article in Lucky Peach and wants to know, you knew you were going to get this one, what's your favorite ramen in Los Angeles?
They make ramen.
I don't know. I actually haven't had ramen in Los Angeles. How often do I come to Los Angeles? Come on, like, this is crazy.
If you're still here
Can you
tomorrow,
say like I don't go to Brooklyn?
Yeah, can you answer this question for us?
Johnny Gold is here. My
favorite ramen at the moment is Tsushita, and it's probably happening in New York too. This Skemen is the future of ramen.
And Tsukemen is sort of the dipping broth. It's a dipping. The noodles are on the side, and you have a very intense broth. I will tell you that my favorite ramen in the whole world right now is Rokurincha in the basement of Tokyo Station. It is a life-changing event. It's so good. I can't even tell you. If you haven't had it, you're in Tokyo. Rokurincha.
Hour in line.
Literally, they serve breakfast ramen, and it's like an hour and a half wait all day. And I don't wait in line for food. I hate it, but I wait in line for refrigeration. So.
Yeah. This is, I suppose, to Peter and David both. Like, one is how do you think Lucky Peach works as a way of collaborating and sharing experiences with chefs all over the world? And secondly, what do you think the goal is and the direction for Lucky Peach in the next few years?
When people ask about the goal of Lucky Peach, I think that it gives us too much credit as in like having a goal for it. I want it to be the Oregon Grinder's monkey of the food magazine world. I just want it to be entertaining and silly and we're finding the best. And like that's what it does. It's just like our way of putting it out there and doing something different and being making entertainment, I guess.
Well, I've known Pete for a long time now, and I think that he finally has a platform where no one can tell him no. And whether it's a story idea, and I think that he has so many great ideas, not just for food, but ideas in general. And it's just a place where we can actually act like buffoons and do what we want to do, and it's fun. So we're going to continue to do it until it's not fun anymore. And sometimes it's not that fun, but it gives, I think, Peter, Chris Yang, and the other contributors room to run where they didn't have before. So.
Well, one of the things about Lucky Peach I think is unusual in the history of food magazines is it's actually chefs talking to chefs about chefing. Not the bogus, so where do chefs go on their night off, or what do chefs, as if they ever have nights off, or what do chefs cook for their families, as if they cook for their families. But the real stuff that you think about day in and day out.
Yeah, I think I've worked around or with chefs for a long time, and one of the hard things, when you're doing real journalism, you have to like quote them and write about them and explain who they are, and we'll run stuff and not explain who somebody is and just try to put it in their language and their voice, because that insider access to chefs has always been an exciting thing for me to have. So I think one of the things we try to do with Lucky Peach is, just kind of put as little of a filter between what chefs are thinking about and saying how we present what they think about it. And, I think it suffers from lack of context a little bit, but hopefully having maybe a more authentic voice from the people that we cover, is appealing to people.
This is kind of an interesting question. What is the real scoop on MSG? And to paraphrase, since there's no scientific basis for anybody's adverse reaction to it, where do you think and why do you think the phobia comes from?
Harold McGee wrote about it in the first issue of Lucky Peach. I think it was 1967.
Yeah, Chinese doctor wrote about it. I really think it's just psychosomatic. I'm sure that there's going to be some outliers and anomalies where people are extraordinarily sensitive to it.
But there was this letter written into the New England Journal of Medicine where this guy asked a question about if MSG caused these reactions. They titled the letter Chinese Restaurant Syndrome, and then the media picked up on that and reported it like it was, facts, when it was really just somebody writing in a question. And then it went on from there. I mean, Chinese Restaurant Syndrome is, like, how catchy is that? That is a great news story. So then you just read, Chinese restaurants, so that's what started it. That's what started people thinking that MSG made them feel bad. But all the published studies about effects, there's no evidence to show that MSG makes you feel bad, unless you're like one of a very small percentage of people who actually have a sensitivity to it.
In a way, it's almost like medicalizing xenophobia.
That, yes.
No, I just think it's this weird.
It just seems,
Studies suggest that
it just seems a little bit racist.
It's a little bit racist.
Yeah.
Not a little bit. I mean, you know, it seems. I always say it's a passive form of racism, but you know, it's really interesting.
That's why he got the.
Yeah.
He's got the Pulitzer.
That's what it is. But like, you know, you talk to some people that are actually Asian, and they tell you that they're allergic, and you know, I don't know. You know, these are not
have food out.
These are not conclusive things. This is just my opinion, and I'm wrong all the time. So, you know, it's just one of those things. But it's something that I don't want to tell a customer. But I think that how should I say? I want our cooks and our staff to actually know what's going on. And it's such a taboo subject that people assume that it's bad food. And just like we're studying microbes, we want to not assume anything that's the status quo. We want to ask why certain things are the way they are. And the more you ask, and the more you study about MSG, the more you realize that while it may not be concrete and like a slam dunk case, there is a lot of evidence to support that it's psychosomatic. It's not really there. And I'm not trying to tell anybody that if they do have allergies that they're wrong. It's just something that I'm trying to learn more about, and it's not definitive, but you know, we'll see. And hopefully there's going to be more studies done, but I would like us to sort of scrutinize this ingredient the same way we do every other ingredient, because that's only going to make things better, I think.
I've wondered myself, there's this thing that's been happening in LA for the last 3 or 4 years, where some of the best new restaurants, especially ones with young chefs, have this little thing on the menu saying, you know, substitutions or modifications politely decline. And does that make its way into Momofuku, or are you happy to change things? Especially since you have knowledge.
Yeah, well, a couple weeks ago, we had an incident where should I say, if it was 8 years ago, it would have been on the menu immediately the next day. But I think I said something along, I wanted to put this on the menu right next to no substitutions or special requests. But if you think that you might have an allergy to MSG, assume that there's MSG in our food. Just so people won't eat our food. It would just not that we don't want to serve them, just we don't want it to be a problem. But you know, it was a healthy debate with all the cooks and the chefs at the restaurant, because we actually want to grow out of not this juvenile stage, but we want to be able to cook for everybody. And we are in the service business. I have to constantly remind myself that. But we got to make people happy. And you know, if somebody says they're, you know, it's just not too offensive, it's too aggressive. And you know, as I said, until it's proven to be true, we're not going to take that stance. So,
how does your approach to creating a recipe differ now from what it might have been before you had the lab, before you were doing it?
Developing recipes now with the lab, before the lab. It's interesting. I think Peter's seen it a lot. It was just a lot of it's teamwork, and a lot of it is sort of, oh my God, we got to put a dish on right now, and it's sort of that urgency to put something on the menu where things just happen. And I'm not saying it's magic, but there's a high failure ratio. But it was unplanned to put dishes on the menu, and now with the lab, as Dan can attest, it's charting our mistakes and our failures. So creativity comes out of failure now, and born out of failure. And that's the most important thing, is that the lab allows us to document just how many mistakes we're making. And you know, that's something that nobody likes to document failure. It just don't. Cooks especially don't. But now that we have to, it really sort of focuses our I wouldn't say creativity, but it focuses on where we should attack.
A lot of, from the students, a a lot of your influence obviously comes from Asia. What are some next level concepts from Asia that you are still exploring or have yet to explore?
I think so much is going to come out of China. We don't even know yet. There's so many types of cuisine in China that I think it's really going to change how we cook. And there's something that's going to shake out of China. Like, you know, right now I'm really curious about the Yunnan province of China, which is, you know, statistically the most diverse population in the whole world, which is saying something. So there's all sorts of stuff that, particularly that area, that I think is really fascinating. You know, they have cheese on the menu, they have tomatoes on the menu. It's really fascinating stuff to me. So hopefully something out there, but you know, I think Chinese food to me is where a lot of new things are going to come out.
Well, yeah, you could be like Dolly and put sea cucumber in everything. Somebody wants to know if they wanted, you've got them hooked, if they wanted more of that pistachio miso. What does she do?
Talk to that guy. Yeah, we have extra. No, we brought a lot with us. If people would like more, bring your cups.
Well, I think on that note, I'd like to thank everyone very much for coming, and especially to our speakers coming from New York. Veronica, who didn't make it up on the stage, but you saw on screen, and David. Making very important, or helping to make very important connections that made this whole science program